Traffic, neurobiologist and health services researcher at the VA, Palo Alto, and the Stanford Psychiatry Department. So immunocytochemistry is a technique where you develop specific antibodies to a protein or part of a protein and then attach labels, so a fluorescent marker or something else that allows you to visualize it so that you can identify where in the body or in a cell a particular protein is at any time.
Okay, and why did you specifically use this?
So we used immunocytochemistry as a way of identifying when and where certain proteins were during certain behaviors. So, for example, we used it to study G-protein coupled receptors that people were developing medication targets for. Though these proteins internalize into the cell, so they sit on the cell surface when they are inactive, and when they're activated, they get internalized inside the cell. And so it's kind of a recycling mechanism, you can think of it as. So when they sit on the surface, a peptide binds to them, and that peptide can't come off of the receptor until it's under different cellular conditions. So you need to acidify the medium. So the cell takes it into a little vesicle, acidifies it there, and the peptide comes off, and then they can recycle the protein back to the membrane. You could basically look in animals to see when they were internalized, what parts of the body or brain that they were internalized, under which behavioral conditions, which would tell you when those receptors were actually active and doing something in the body. We were studying neurons in the spinal cord and brainstem for the most part, because we thought that the receptors we were studying, the neurokinin 1 receptor, was involved in pain processing. We just wanted to use it as a marker for when the receptors were being activated. We could test drugs to see which drugs were blocking the activity. This is one of the methods that we used. Because to some extent, when you give drugs systemically, which is the way people mostly want to, you can't give them at whoppingly high doses. So you don't really know exactly to what extent they're stopping activity. So it provided an activity marker. It also, but for the most part, it helped us figure out when those receptors were active in the first place, because it turns out that they're only rarely used. They're sort of an emergency response system.
Okay.
Like chopping your arm off, or like, having an inflammatory injury that makes your ankle swell up like this and then poking it, or sticking it in boiling hot water or something like that. So it's not something you take for a headache. You want to target for a headache. It's something only for really serious chronic conditions.
So
it turned out that they really weren't very good painkillers, and so we wanted to figure out why they weren't working, what they were really doing. They seem to have they're really good for nausea, it turns out, and some other sort of more psychologically relevant stuff. So saporin's a cellular toxin that only works when it's inside of a cell. So if it can get inside a cell, it'll kill it, but if you just poured it on a cell, it wouldn't do anything. So we conjugated it to a neuropeptide, same neuropeptides that I've been studying before with immunocytochemistry, in this case substance P. It's a neuropeptide. It's created only in certain subpopulations of neurons and released. So in the spinal cord, for example, where we were studying it, it's released only under severe injury states. So we link those two together, and then you could inject that when you inject that into animals, the substance P will specifically bind to its own receptors, and the neurokinin 1 receptors that we talked about before. And because those receptors internalize when they're activated, when the substance P bound with a saporin attached, it would get sucked into the cell. The saporin would then kill the cell, right? So you could selectively lesion out specific populations of neurons with that neurokinin 1 receptor. So we thought if you could lesion out those neurons that were getting the signal about pain, that you'd stop transmission of those severe chronic pain chronic pain information up to the brain, so you'd never know what happened. It did. Yes, it did. So, I mean, you could lesion out. It definitely lesioned out the cells. Most of the cells in the dorsal horn and the spinal cord that had the neurokinin receptor, and it did result in reductions in hyperalgesia and allodynia, so excessive pain after inflammatory and nerve injury in animal models. I don't think it made it very far in the problem being you had to get it directly on those neurons and not on other neurons that had substance P, and there are the neurokinin 1 receptor, and there aren't that many neurons that have the neurokinin 1 receptor, but motor neurons also do. Theoretically, yes, though. You wind up with muscle weakness and things like that as you lesion out motor neurons. One of the things we find is that cells were dying because they were running out of energy, basically. So we wanted to find ways of boosting their ability to take in energy, and hopefully that would keep them from dying. So we worked with virologists in the lab who developed herpes simplex virus that contained the glucose transporter gene.
Oh, okay.
So a gene that would help the cells take in more glucose when they were under energetics challenges. So basically started with HSV1, the virus that causes cold sores and such, and took out parts of it that were required for replication, added back in genes for the glucose transporter gene, leaving us with a virus that could not replicate but could infect a cell and bring in extra copies of the glucose transporter gene. We found that when we did infect neurons with this, you could in fact improve their energy states, so that neurons were healthier, they were doing better when you subjected them to challenges like stroke and seizure, and it did wind up having some neuroprotective effects. If you pre-treated the animals with the virus, then you could get you could save neurons following these insults, and if you injected them with the virus very shortly after that.
No.
They didn't last that long. They'd last for a while, like on the order of months, but not
and then
for decades. You have to basically like run somebody to the emergency room and do neurosurgery. Not exactly the best treatment, but potentially workable.
And so you had some success or not?
Yeah, no, they did, and they went to clinical trials.